Thank you so much. Thank you for the kind invitation. And are my slides, are they in the correct form?
They're great.
Perfect. Okay. So I'm going to tell you a little bit about what my group has been doing, kind of more on the biomaterials side of things. And so to give you a little bit of background, I actually did my PhD work in traditional hydrogen oxygen fuel cells and kind of developing electrocatalysts that would sort of minimize the use of platinum and platinum group metals. So I have a background in electrocatalysis, and for, I guess, about the last 20 years, we've been using that background to transition from traditional electrocatalysis to bioelectrocatalysis in fuel cells. And really, if you think about the difference between a fuel cell and a biofuel cell, the only difference is that you're exchanging what is typically a metal nanomaterial catalyst at the anode and or the cathode with a catalyst of biological origin. And you might say, why would I be interested in using a catalyst of biological origin? And the real reason is really just to open yourself up to a much wider variety of fuels. So if you think about a fuel cell and you compare it to a living organism, the sort of fuel is basically everything that we eat. So we consider it food. In the fuel cell world, we consider it a fuel. And so if you think about it, hopefully, whether you're having breakfast, lunch, or dinner right now, hopefully you had an opportunity to have a little bit of carbs, a little bit of protein, a little bit of oils, a little bit of fat and protein. And you have the metabolic pathways within you to break those down. So biocatalysts in comparison to, say, platinum catalysts are really good at deeply oxidizing really complex fuels. And so that's actually how I got interested in biological fuel cells, because pretty much anything you can think about yourself or a microorganism metabolizing, you can think about using as a fuel. So this allows you to continue to think about a lot of the alcohol-based fuels that we talk about with fuel cells, but it also opens you up to, thinking about carbohydrates, thinking about oils. We've, made fuel cells that will oxidize JP8, so jet fuel, because essentially there are microorganisms that are used to clean up waste spills, so oil spills. And we can use those metabolic pathways and those microorganisms to actually oxidize, really complex fuels. So that's kind of the good part of biofuel cells. The bad part of biofuel cells is that when you think about a traditional catalyst, if you think about a small platinum nanoparticle, it's a small platinum nanoparticle. It has many active sites. So it could be, on the order of, say, 2 to 5 nanometers. So similar size to the proteins that I'm going to talk about today, but it has many active sites. And the particle itself is conductive. So it can conduct electrons to and from your current collector very easily. If we think about those proteins that are responsible for metabolism, they are not conductive. So you have an active site, a single active site for every kind of 5 nanometer particle. You've got one active site, and that one active site is sort of buried in a protein or peptide structure that is not conductive. And so that means that really the challenge in biofuel cells is being able to get the electrons in and out. And there's really 2 main strategies to do that. The first strategy is direct electron transfer. Can I orientate that protein so that my active site is within tunneling distance? So can I get my active site to be within a nanometer of the electrode surface to be able to tunnel electrons effectively? And this is quite a bit of a challenge. There are certain proteins that are typically small and can easily do direct electron transfers, but a lot of proteins can't do direct electron transfer, and we have to instead sort of have mediation schemes to be able to mediate those electrons to from the electrode surface. And that's really what my group does a lot of, is the sort of materials electrochemistry to design materials to help biological entities interface with electrode surfaces. So we design things like redox polymers that can do self-exchange-based conduction of electrons to the electrode surface. We functionalize or design nanomaterials that can orientate proteins so that they can do direct electron transfer. We make all kinds of sort of mimics of cellular membranes to be able to keep membrane proteins happy at the electrode surface. And we do this for a wide variety of different applications. So some of those are in biosensors, and that's probably the largest application of bioelectrochemistry out there. So all the glucose test strips that people use who are testing their blood glucose, diabetic patients testing their blood glucose, all of those are sort of bioelectrocatalytic systems. So we do a bit of biosensor work. We do a bit of work on energy conversion. And more recently, we're doing quite a bit of work in electrosynthesis. What I'm going to talk to you today is sort of the energy side of things, and really kind of how you can take these biological nanocatalysts and actually get them to function on electrode surfaces for energy conversion devices. And so if you think about this, most of the metabolism in a living organism, those metabolic pathways are basically based on hold on, let me get my pointer to work here, sorry. Are based on NAD-dependent dehydrogenase enzymes. So most of the oxidation that happens in metabolic pathways actually require a cofactor NAD that actually binds with the substrate or the fuel that's being oxidized. And when you oxidize the fuel to its sort of first product, you take that cofactor NAD and you form NADH. And so really then you're using that NADH as a mediator to mediate those electrons and sort of regenerate that cofactor NAD. And so if you look into the literature, people have been basically developing electrodes that are NAD or NADH based for about 40 years at this point. Myself, only about 20 years, but historically people have studied a lot of these NADH systems. And the thing that they're challenged by is the fact that if you look at NADH electrochemistry on most inexpensive current collectors, so things like carbon paper and carbon cloth, you have overpotentials for NADH or NADH oxidation that range from 500 millivolts to a volt. So if you think about a fuel cell as a 1.2 volt battery for all intents and purposes, if you lose a volt of that to the overpotential of NADH oxidation, then okay, yes, you have a mediation scheme, but you're not going to have a very useful battery. And so we have to add an additional electrocatalyst here to actually catalyze that NADH oxidation to regenerate the NAD. Methylene green, which is a phenothiazine that has actually been discussed for redox flow batteries, is also a well-known electrocatalyst for NADH, and it's very easy for us to polymerize it directly on an electrode surface. So we can take a carbon paper or carbon cloth electrode, place it in a solution of methylene green in a borate buffer, and as we scan, I will tell you that I'm from the electroanalytical community in the US, so this notation is polarigraphic notation and not IUPAC notation, so I apologize if you might have to look at it upside down and backwards. But as you start scanning in the positive direction in that solution of methylene green, you'll adsorb a first layer of methylene green, and then at potentials past 0.8 volts, you'll polymerize it. Then you can scan back, deposit another layer, polymerize it. Scan back, deposit another layer, and polymerize it. So you can do a layer by layer deposition of this conducting polymer that will act as an electrocatalyst for that NADH oxidation. So if you think about it, we're basically taking carbon cloth or carbon paper, we're coating it with an electrocatalyst, and then we're coating it with one or more enzymes. And that's really what I wanted to talk to you about, is sort of the enzymes and the placement of the enzymes on the electrode structure to actually allow us to have efficient bioelectrocatalysis. So as I said, hopefully all of you have had breakfast, lunch, or dinner, depending on what time zone you're in. And so you within yourself have a series of metabolic pathways that are breaking down and completely oxidizing whatever it is that you consume. So if you're consuming sugar and that sugar is going to be oxidized by the glycolytic pathway into something like pyruvate, and pyruvate will feed into the Krebs cycle that is in your mitochondria to completely oxidize that pyruvate to CO2. So metabolic pathways are excellent and very efficient at deeply and completely oxidizing a wide variety of fuels. You don't have to just have carbs. We actually don't want you to just eat carbs. you can have a completely balanced meal, and as you eat a completely balanced meal, you have metabolic pathways to break those down. And so, you know, early on when we got interested in biofuel cells, we were interested in whether or not we could actually get these metabolic pathways to function on electrode surfaces so that we could actually use them for energy harvesting. And so you might think I'm going to talk about glucose or sort of sugar fuel cells, but I'm actually going to start with pyruvate fuel cells, mainly because the glycolytic pathway is electrochemically boring. So if there are any biochemists in the audience, it's biochemically beautiful, but it only has one oxidoreductase or one electron producing enzyme. So it's electrochemically boring, whereas the Krebs cycle has 5 oxidoreductase or 5 electron producing enzymes, and is therefore sort of much more interesting from that perspective. And so we wanted to see, okay, can we take a metabolic pathway and get it to function on an electrode surface and show that we can use enzymes to deeply oxidize really complex fuels. And so what we did was take the Krebs cycle or the citric acid cycle and look at it in terms of what was out there in the literature. And so pyruvate is the main substrate for the citric acid cycle. And there was actually research in the literature where people had taken pyruvate dehydrogenase, immobilized it on the electrode surface, and shown that you could get 2 electron oxidation of pyruvate. You also can get 2 electron oxidation of pyruvate on some platinum alloys. So that's not particularly impressive all on its own. So what we wanted to see is, could we add both electron producing enzymes, the enzymes here in red, as well as the non-electron producing enzymes of the Krebs cycle to actually get this full pathway to function and completely oxidize pyruvate to CO2. So we decided to sort of make electrodes where as we made additional electrodes, we were titrating in additional enzymes to more deeply oxidize our pyruvate fuel. And so our fuel cell looks a lot like a traditional hydrogen oxygen fuel cell. So you basically have that MEA structure where you have a separator here, Nafion separator. Our cathode is an air-breathing sort of platinum cathode, but now on our anode we have carbon paper. That carbon paper is coated with our electrocatalyst to regenerate the cofactor, and it's coated with one or more of the enzymes of the Krebs cycle, where we start with just a single dehydrogenase, then we add a second dehydrogenase and its corresponding non-dehydrogenase until we have the whole pathway functioning. So we basically made 5 different fuel cells here, or 5 different anodes at least. They all have the same sort of separator and cathode, but 5 different anodes where we had 1 dehydrogenase, 2, 3, 4, and 5 to try to completely oxidize that pyruvate. And when we do that and we look at the power curves, it's quite interesting because we're titrating the same catalytic activity of the next enzyme down onto the electrode surface. And so you'd expect, as we go from 2 electrons to 4 electrons, we would double our current, and since power is current times voltage, we would sort of double our power. So we would see these sort of doubling, tripling, quadrupling of our power curves. And when you look at the power curves, you can see down here in pink when we just are doing that 2 electron oxidation of pyruvate. When we go from pink to blue, we see an increase in current. Then from blue to yellow, another increase, but we're not quite doubling and tripling until we add this final enzyme, malate dehydrogenase, to the system. And we see this huge 26-fold increase in current density and power density. And this is a perfect example of doing research and expecting to see things double, triple, quadruple, etc. And then you suddenly see this 26-fold increase, and you're like, something must be wrong. We have to go back to the drawing board. And in actual case, nothing was wrong with the experiment that we were doing. It was just naive of us as electrochemists to not really fully understand the biological system. So unlike a chemical system, biological systems have built-in metabolic control. And that built-in metabolic control means if you build up any one of these intermediates, that intermediate will actually inhibit previous enzymes of the cycle. So in order to keep metabolism from going awry and keep life functioning, the metabolic systems have this built-in metabolic control where all of the intermediates actually inhibit other enzymes of the pathway in order to maintain that control. And so if we start to make an artificial pathway that doesn't have the whole cycle, we start to build up intermediates, and those intermediates actually affect this initial enzyme, pyruvate dehydrogenase. And so it's not till we add the final enzyme and we actually have this cycle functioning, then we aren't building up intermediates, and we see higher current densities and higher power densities. We've applied this concept to other metabolic pathways or even other substrates, like for instance, adding additional enzyme lactate dehydrogenase to oxidize lactate. And we see much the same system. If we have a natural metabolic system, it has built-in metabolic control. And we see that over and over and over again. Some things to think about when you're thinking about biofuel cells is that we oftentimes talk about energy density or specific energy. And in the case of a biological fuel cell, it's not just the energy density of the fuel, and it's not just how many electrons you can get out of the fuel, but it's also the solubility of the fuel, because these have to operate in an aqueous environment. So if we look at the two examples I just showed you, pyruvate and lactate, pyruvate's not very soluble. It's only soluble to about 1 molar. And so if you look at theoretical energy densities, even if you do complete oxidation, they're still pretty low. Where if you look at lactate, lactate's extremely soluble, and you can get really high energy densities from completely oxidizing it. So the moral of the story is you need complete oxidation, but you also need to have high solubility to be able to make energy conversion devices that are ideal. Now, I told you that the Krebs cycle or the citric acid cycle exists in the mitochondria. And I'm an analytical chemist, an electroanalytical chemist, but an analytical chemist by training. And that means we spend a lot of time thinking about what would be good control experiments and what are good blank experiments, and really sort of thinking about experimental design. And so as we started to take all of these purified enzymes and make these electrodes, we started wondering if we couldn't use mitochondria as a control experiment. So in theory, mitochondria has evolved to have all of the enzymes of the Krebs cycle and all of the enzymes at the right concentration to have high flux through the pathway. So it efficiently takes pyruvate and completely oxidizes it to CO2. So we were like, can we actually just lyse the mitochondria? They have all the enzymes in the right concentrations and use that as an experiment. And so we actually made sort of mitochondrial fuel cells. And we basically made 2 different types of mitochondrial fuel cells, one with lysed mitochondria, one without lysed mitochondria, so intact mitochondria. And if you think about this from electrochemistry perspective, anytime you have to a resistance to transport, like the transport of pyruvate over a mitochondrial membrane or across a mitochondrial membrane, you would expect to see kind of decreases in current density associated with that sort of resistance to mass transport. So we actually expected to see sort of very little response for the intact mitochondria because of that resistance to transport of the fuel across the membrane, but seeing sort of really ideal response associated with the lysed mitochondria because we didn't have that resistance to transport, and we also had all of the enzymes kind of in their right concentration to be able to have high flux. But that's not what we saw electrochemically at all. So we actually saw the opposite of that, where the lysed mitochondria had a very small response. The whole mitochondria had a really large response. And at this point, you again go back as an electrochemist and say, okay, you know, biology is confusing. When you sort of go back to that biology, you have to think here, there's something special about whole mitochondria. And that's something special about whole mitochondria when you go into the literature is the fact that Paul Schreier at the University of Texas had proposed back in the late 80s and early 90s that in order to have the fluxes through metabolic pathways that we have, and remember from an electrochemistry perspective, fluxes are current density, in order to have those sort of high fluxes through metabolic pathways, you couldn't have the individual enzymes by themselves. They had to form super complexes. And he proposed that these super complexes existed and that they actually formed channels between active site 1 and active site 2 and active site 3. So not only are you putting active sites close together so that you have sort of very small distances to diffuse across or to transport across, but also you had little tunnels that were allowing you to ensure that your intermediate was going from enzyme 1 to enzyme 2 and not finding enzyme 4, 5, 6, 7 by mistake. And so he proposed this, that we didn't really have the structural biology techniques at the time to be able to prove that this was indeed in a structure. So when we got interested in this, we had to decide to go into a little bit of, do a little bit of structural biology ourselves. And we actually took his sort of proposed structure, which is the dark system here, the dark figure. And we went back and actually used current structural biology techniques, so cross-linking mass spec techniques, to actually find out that he was indeed correct. There was a metabolone. There are tunnels that go between active site 1 and active site 2, but there are electrostatic patches, and since our intermediates are all negatively charged, these positively charged electrostatic patches actually keep the intermediates from going from one active site to the next active site to the next active site. So we want to see if this could actually be useful to us electrochemically. So we actually took Saccharomyces, we took yeast, we isolated their mitochondria, so little orange dots here are the mitochondria. We treated the mitochondria with homobifunctional cross-linkers to cross-link that metabolone in place. We lysed the mitochondria, isolated the metabolones, put them on electrode surfaces, and actually showed that we do see much increased current densities and power densities associated with having the metabolome. So the metabolome is important, and biology makes this metabolome all on its own. We needed to figure out how we could make the metabolome sort of outside of this system. So we were interested in kind of combining biomaterials and nanomaterials to see if we could artificially form a metabolome. And so we use a lot of carbon nanotubes for our electrode structures. So we took carbon nanotubes and there's sort of single-stranded DNA that will wrap itself around carbon nanotubes. And then we actually could hybridize a second portion of that DNA and actually have regions of the DNA that will actually bind zinc finger domains on proteins. So we took 2 enzymes, alcohol dehydrogenase and aldehyde dehydrogenase, that are responsible for oxidizing alcohols to aldehydes, and then aldehydes to either acetate or formate, depending on whether you started with ethanol or methanol. And with the help of Scott Banta at Columbia University, he functionalized these proteins with zinc finger domains that would actually bind specific double-stranded DNA regions. And we could change our spacers, so we could change how close or how far the proteins were apart. But the hope was that we'd be able to design an artificial metabolome that would form directly on the electrode surface. So what we did was basically take the carbon nanotubes, a really long strand of DNA, only part of which will be hybridized, sonicate them together so that the DNA winds around the carbon nanotube, hybridize with that second short tier that has sort of 2 binding components and a spacer, cast that on our electrode surface, place that in a solution that contains the proteins with the zinc fingers on them, and it should self-assemble into a structure. We then have to make sure that it can still do catalysis, and we did indeed show that it can still do catalysis. And when we get done, we can actually see that when we have that DNA forming that metabolome, we have significant higher currents than if we don't have the DNA. So indeed, the performance is enhanced by us being able to sort of develop this metabolome on the electrode surface without having biology do it for us. Now, these are everything I've shown you to date has been all natural metabolic pathways. And one of the things that we're really interested in is designing non-natural enzyme pathways. So if you think about a fuel like glycerol, glycerol is pretty high in energy density. It's a byproduct of biodiesel production here in the US, which makes it pretty inexpensive. It's low in volatility. But if we were to oxidize it through its natural metabolic pathway, it feeds into the side of the glycolytic pathway and through the Krebs cycle. And 21 enzymes later, you can completely oxidize glycerol. I have some really fantastic graduate students, but none of them wants to figure out how to find a way to get 21 enzymes to be functional on an electrode surface. And so we want to think about non-natural enzyme pathways that really just do the things that an electrochemist cares about. I want to break carbon-carbon bonds and I want to get electrons. I don't need to phosphorylate something and a couple steps later dephosphorylate it. So there's a lot of biology that's there that has a function in biology, but it doesn't have a function in electrochemistry. And so we've looked at ways that we can use promiscuous enzymes to be able to make non-natural minimal enzyme pathways. And we've done that for fuels like ethylene glycol and glycerol, where we've taken some alcohol dehydrogenase and aldehyde dehydrogenase and oxidase enzymes that are quite promiscuous, where the alcohol dehydrogenase will work on all three OH functional groups. The aldehyde dehydrogenase will work on 2 aldehydes. Oxalate oxidase will cleave both of those carbon-carbon bonds. And we can, with 3 enzymes, completely oxidize glycerol to CO2. These promiscuous enzymes are also heme-containing enzymes. So these enzymes were actually all isolated and purified from bacteria growing in red wine. And so they're really rich in iron. And so they have a lot of hemes that act as internal relays within them. So not only did they not evolve to be specific like you normally think of an enzyme, but they're also sort of very heme-rich, which allows them to do direct electron transfer very readily. So it's very easy for us to make electrodes out of them. The two of these enzymes, oxalate oxidase and the PQQ-dependent alcohol dehydrogenase, are both phenomenal enzymes to work with electrochemically, but the aldehyde dehydrogenase is not. And so when you start to think about developing enzyme pathways and non-natural enzyme pathways, your first thought goes to Frances Arnold and all of her work with directed evolution. And so we started trying to do directed evolution to evolve aldehyde dehydrogenase to be basically do the chemistry that we wanted it to do versus its natural system. So unlike what Frances usually does, which is directing something to be more specific, we were actually trying to do directed evolution to make it less specific. And we were just really struggling at this. And I went to lunch with one of my colleagues, Matt Sigmund, who's an organic chemist, and he said, Shelley, I don't understand, why are you trying to evolve? Why are you trying to go against nature? Nature's made these, more specific over time. Why are you trying to make them more promiscuous? You should just be using an organic catalyst because they're very promiscuous. And then you won't sort of have this issue. And it's like, well, all of the organic catalysts that you work with in organic solvents. My other 2 enzymes are not going to like those organic solvents. And I need to get these catalysts to be essentially heterogeneous catalysts on the electrode surface. And so he showed up in my office the next day and he's like, this is your catalyst. He's like, you should give Tempo a try. It will catalyze both alcohols and aldehydes, so eliminate 2 enzymes. And it works in water. And you spent, your life figuring out how to design electrodes to put things, to immobilize things on electrode surfaces. Surely you can immobilize Tempo on the electrode surface. So we actually gave this a try. Tempo itself did not work, but Tempo with an amine functional group did work to do this complete oxidation process. And then we started thinking about how we could, for all intents and purposes, heterogenize an organic catalyst onto the electrode surface. So we took linear polyethyleneamine. We functionalized linear polyethyleneamine with Tempo to make a redox polymer where basically the tempos are close enough to do self-exchange-based conduction. We can then take this polymer, cross-linker some carbon nanotubes and the enzyme and cast it on an electrode surface. And we do this, we compare the monomer to the polymer. You can see that having it on the electrode surface is really important to having high current density and high power density. And when we looked at all the different sort of substrates that you might want to work with, Matt was absolutely correct that it is quite promiscuous. It can work with methanol and ethanol and glycerol and carbohydrates, both monosaccharides, disaccharides, polyols. So it's very promiscuous from that perspective. So this is giving us a chance to go into looking at hybrid systems where once we get to a situation where we're trying to make that non-natural enzyme pathway, some things just don't do enzyme engineering well. And we can then think about replacing those either with an organic catalyst like I showed you here or with an inorganic catalyst as well. So hopefully in the last 35 minutes, I have shown you that enzymatic biofuel cells allow you to use a wide variety of fuels. I showed you pyruvate and lactate and alcohols and glycerol. but that electron transport is more complex because you don't have a conductive catalyst, and therefore you really have to think about your materials design at your electrode surface. Hopefully I showed you that metabolons are indeed important. And we've got to think about, again, materials innovation to be able to develop electrodes that actually form those metabolones for us. And that every once in a while you can't enzyme engineer what you want. And that's a great opportunity for you to use hybrid catalytic cascades to do the chemistry that you want. And with that, I'd like to thank the funding agencies, my collaborators, and my research group, because I am just the talking head. And they're the ones who did everything that I showed you today. You guys for being a great audience, and I would be happy to answer any questions you have.
Great, thank you, Charlotte. This is fascinating. So you're basically reducing the biological entities to its bare essential minimum to do these catalytic actions. So any questions from the audience? So let me start. So when you talk about the hybrid catalyst at the end, this is a great idea. So have you considered a hybrid between the enzyme, artificial enzyme with inorganic, right? Because you are in need of an electron conductor anyways.
Yes. Yes, we have. So we have a collaboration with Plamen Atanasov, who used to be at the University of New Mexico and is now at the University of California, Irvine. And so we have been sort of combining some of his nanocluster catalyst work with our enzyme systems. So yes, I think, you know, the struggle with organic catalysts is that most of them operate solely in organic solvent. And the challenge with sort of a lot of the more traditional nanomaterial catalysts is they either operate in highly alkaline environment or highly acidic environments. And so if you think about the enzymes, they're not going to handle those harsh environments. So you have to find catalytic systems that will operate not necessarily at neutral, but will operate kind of in the pH 3 to 11 range.
So what do you think would be the, I'll say, the entry application point for this biofuel cells?
Yeah, so I think, you know, if you look at our continued interest kind of in wearables, so our Fitbits and sort of fitness monitoring, I think energy harvesting is likely going to be sort of one of your first sort of low entry areas. For a number of reasons. One, you actually have quite a bit of some of the fuels that I talked about, like lactate that are there in sweat, as well as the fact that it allows you to sort of combine something that might be fitness monitoring with also something that may be sort of chemical monitoring for people who are sort of looking for biomarkers.
I see. Great. Right, so I have a question from the other platform. So the question is, they are wondering about the stability of this biocatalyst, and is there any way or approaches to improve their recycling use? Like, can you recycle and reuse them?
Yeah, so that's a really, that's a very good point. So if you take an enzyme and you just put it in solution and let it sort of sit in solution, it's not going to be stable for very long. It's probably going to be stable maybe 2 or 3 days, and proteases will start consuming it, and it will denature and be non-functional. So it's really important to actually sort of encapsulate that protein on the electrode surface so that proteases can't attack it and that it can't sort of easily denature. So again, that kind of goes to thinking about materials, whether those are, you know, sort of mesoporous materials or polymer materials that can help you to do that sort of protectiveness. So with the stuff that I showed you today, if you look at the Krebs cycle enzymes, one of those enzymes is a membrane-bound protein, so succinate dehydrogenase. And of course, you're always limited by your weakest link, and that's the weakest link. So those particular fuel cells are only stable for a few days, where the sort of glycerol system that I showed you is stable for months because it's actually protected in a polymer, and they're not they're much more stable proteins themselves. And so there's a lot of work right now, people looking at extremophilic microorganisms and the enzymes that come from them as enzymes that would be stable for very long periods of time.
Great. Let me check the chat box. So meanwhile, for anybody who's here in the Zoom meeting room, feel free to unmute yourself and just ask away. I'm checking my box here. So while I'm looking, Shirley, so I came across what people are doing this drug discovery when you talk about the encapsulation with the polymer shells, right? So there has been work on when you encapsulate one copy of protein or multiple copy of protein in there. I'm just wondering, has this been explored in the biocatalysis field? So when you talk about metabolons, right? So have you tried to encapsulate those in a kind of a vehicle?
Yes. So there has been a lot of work in different strategies for enzyme immobilization. Some of the strategies of drug discovery work, others do not, because the thing that you have to think about when you're putting it on an electrode surface is that you have to have sort of facile ion transport. So things that would be like ionomers tend to work better than something that is not going to allow ion transport as easily.
I see. So we need to have, I guess, diffusion, right? Kinetics needs to be taken care of.
Yep.
And a lot of, and the catalytic activities. It's very interesting and very challenging field, I have to say. So if there are no more questions from the audience, let's thank Sherry again for a great talk on biocatalysis.